Okay, good. This presentation is very long, so I will skip some of the parts, but you will get the slides that there's much more detail than you probably want to know about, but just for curiosity, and again, if you are interested, you can ask me tomorrow or on Thursday. So it's MAXOCC lecture, but this protocol that we set up that we call maximum occurrence, MAXOCC in short, is based on using paramagnetic NMR data together with SACS data, as I mentioned already. So we have first to spend some time defining what paramagnetic NMR data means. So, of course, you record what we call paramagnetic NMR data in systems that are paramagnetic, such as some metalloproteins, where the peculiarity is that they contain metal ions, in this case, which have unpaired electrons. So because they have unpaired electrons, there's an electromagnetic moment, and the electromagnetic moment is typically much larger. So the moment associated to the electron itself is much larger than what you get associated with the single nuclear spin by a factor that is more than 600. And being all magnetic moments, of course, there's an interaction between the nuclear magnetic moments and the electron magnetic moment, and we mentioned this already in the first lecture. And so you have an energy of interaction, and for example, that contributes this kind of interaction contributes to relaxation rates, which is what I mentioned earlier. Then there's a so-called paramagnetic susceptibility. That's actually the parameter, this chi value here, the paramagnetic susceptibility per molecule, is the parameter that gives you the proportionality between the electron magnetic moment and the applied magnetic field. So it basically takes into account the splitting between spin levels, in the splitting in energy between spin levels that you obtain when you apply a given magnetic field. You get polarization along the z-axis, the magnetic field axis, and the proportionality constant is chi divided mod 0. So chi tells you how much electromagnetic moment you obtain given the magnetic field. Electron spin or electron magnetic moment has the peculiarity that it can interact with the orbital magnetic moment. So the electron, as you know, is sitting in an orbital on the metal atom. And if there's an orbital magnetic moment, you will get an interaction between the orbit and the spin, what we call the spin-orbit interaction, by the way. And this makes chi anisotropic. It means that the susceptibility, so the magnetic moment due to the electron spin that you obtain, is different with different orientations of the protein with respect to the magnetic field, as it says here. So if you change the orientation of the protein with respect to the magnetic field, you get a different value of chi and basically different, you apply a different projection with respect to chi, and therefore you obtain a different magnetic moment. This is important because when you are looking at nuclei, as we do in NMR, the electron-nucleus interaction contains the susceptibility. And if the susceptibility is not isotropic, you get an additional contribution to the chemical shift that is called pseudocontact shift. So this so-called pseudocontact shift is entirely due to the fact that there is an anisotropy of the magnetic susceptibility of the electron, which is given by these two numbers here. this value is the difference between the magnetic susceptibility along the z-axis minus the average in the plane, and then there is the rhombic anisotropy, which is the difference between the two in-plane components of the magnetic susceptibility tensor when the Cartesian reference frame is coincides with the frame in which this chi matrix is diagonal. So basically you have to take a reference system which is the one that makes the susceptibility matrix diagonal, and then you can use these equations. And as you see, you get a formula for the pseudocontact shifts that depends on the spherical coordinates of your nuclei with respect to the frame of reference, and it also depends on the distance between the metal and the nucleus under observation. You can draw, you may have seen some of these pictures in papers or in books. You can actually draw what we call iso shift curves, so where you see, where you plot regions of space that are sitting on this surface where the value of pseudocontact shift is constant in absolute terms. But actually you see that you have a positive region and a negative region, and that the shape of this thing depends on the value or on the magnitude of the rhombic anisotropy with respect to the axial anisotropy. So these pseudocontact shifts are proportional to r to the third and depend on angular parameters when you fix the reference frame as the frame in which this magnetic anisotropy matrix is a diagonal matrix. This is an example for a real protein where we measured these values, and you have positive shifts here and negative shifts in the rest of the protein. And actually, the directions in which this susceptibility anisotropy is or diagonal are not random, but they are determined by the coordination environment of the metal ion. So there's some inorganic physical chemistry that is going on here, which we don't care much of today. So for example, in this system where you have the iron ion which is sitting in a planar porphyrin, sorry, it's coordinated by a planar porphyrin and two axial protein ligands, which are not shown here for clarity, you actually get a Z, because of symmetry reasons, local symmetry, you get a Z-axis which is normal to the porphyrin plane, and that's where you get the positive values, and then you get negative chemical shift, sorry, pseudocontact shift values in the plane of the porphyrin. And there's actually an angle that you can more or less see here, where the negative, so to say, effect of the in-plane component and the positive effect of the axial component balance out, and this is the so-called the magic angle for pseudocontact shifts, which is analogous, for those of you who are familiar with solid-state NMR, of the magic angle of in solid-state NMR. If you have a system where you can bind different metal ions, such as these calcium binding proteins, which are a class of proteins which we've been heavily investigating in Florence, you can substitute the calcium ion in the protein by different metal ions of the Lanthanide series, which have different magnetic susceptibility with respect to one another, and so you get different effects both in size, that is the size of the spheres for the same atoms. So you see this Lanthanide is inducing much larger shifts than this other one, but also you get a difference in sign of the pseudocontact shifts. So here you have negative values, here you have positive values, because the spin-orbit interaction is different in the different lanthanides. Now, if you have this magnetic susceptibility anisotropy, as I was mentioning before, you get different magnetic moments depending on the orientation of the protein with respect to the magnetic field, and so also the energy of interaction between the protein magnetic moment and the magnetic field is different. So basically, you have different energies depending on the of the protein in the magnet, depending on how it is oriented with respect to the z-axis, which means that you don't get an equal population of all the orientations, but you actually have favored orientation with respect to the field. And this is self-induced, so at variance with what we described before, where you had this alignment media and the protein was orienting because of interaction with the molecules that were in their turn oriented in the NMR tube. Here the protein is orienting by itself spontaneously against the field because of these paramagnetic effects. And you get, because of this, you get again measurable residual dipolar couplings. The difference with respect to before is that now the residual dipolar coupling that you are measuring is not longer dependent on the anisotropy of motion, the D parameter that was in the former presentation, but it's actually again depending on the magnetic susceptibility of the system, which is, if you do your measurements properly, by subtracting as you remember, you have to subtract oriented versus non-oriented couplings to measure this residual dipolar interaction. Here you get the same result by subtracting paramagnetic and diamagnetic systems, and in this way, I think it's on the next. No, it's not. Remove it. In this way, you get residual dipolar couplings that, in principle, depend on the global, so protein plus metal magnetic susceptibility anisotropy. But if you use a diamagnetic system as the reference, so you subtract that, you also subtract the effect of the protein, and so these two numbers should be exactly the same. So you get the same delta chi values, axial or rhombic, for pseudocontact shifts or for residual dipolar coupling. So we are measuring the same thing with two completely independent kind of measurements, because this just comes from your assignments and this comes from the measurement of, for example, J1, 1J couplings. And you see the similarity here. The residual dipolar coupling does not depend on the distance to the metal, but if you are measuring, for example, the amide proton, amide nitrogen residual dipolar coupling, it just depends on the bond length of the amide bond, which is a constant. So in principle, all this thing here, if you're working at fixed magnetic field, is constant and does not depend on the metal to proton distances or metal to nucleus distances in this case. So pseudocontact shifts have a reference of frame with a given origin, that is where the metal ion is, whereas the residual dipolar couplings, let's say, are function of the same coordinates in terms of angles that are distance independent. And this is important, will become important later, actually. So this is what I was mentioning about cancelling out the effect of the molecular, of the diamagnetic protein frame. If you subtract the splittings, or if you subtract the chemical shifts in a paramagnetic and diamagnetic, otherwise identical system, so for example, which for example you can produce by reintroducing calcium, which is diamagnetic, in the protein. So you measure the shifts, you can do the subtraction, you obtain the pseudocontact shifts, you measure the couplings for the paramagnetic proton and the normal diamagnetic proton. You get the splittings, and they relate to the same parameters, that is the electron magnetic susceptibility and isotropy. Okay, this is just to say. And you can use these things in calculation. I had data also to show that, but I guess we ran too late. So if you want to see, I will include, for example, pseudocontact shifts in the Explorer Amber calculations. I'll be happy to show that to you tomorrow. If you just I mentioned, if your protein does not contain any metal binding site, there's a lot of literature now that you can actually, for example, chemically modify your protein. So you express your protein, then you can, there's now a wide portfolio of chemical reagents that you can use to modify the protein and get a cage or a chelating agent attached to the protein that will bind tightly the metal ion. So even if it's not paramagnetic, it's relatively straight, or it's relatively simple chemistry to introduce the metal ion there. So what the aim of today is to show how these things are useful in terms of characterizing flexibility within large multi-domain proteins, and our PET system for that, it's vertebrate Calmodulin, which is a two-domain protein. Each domain contains 4 helixes, and each helix, turn helix motif, binds a metal ion. Sorry. So you can, the entire protein is capable of binding up to 4 metal ions, calcium ions when it's in the cell. And it's actually split into two domains, so-called EFNs. So each of the domain is made by 4 helices and can bind to metal ions. And they are connected by a very flexible linker, and there's data from the 90s again that shows that this flexibility is relevant to the physiological function as well. So these are the two conformations, the two limiting conformations. Let's say there's an extended conformation where the linker is actually folded in as an helical structure, and so you get this very long helix that is connecting tightly the two domains. But actually, you can show, and there's data showing that's NMR measurements showing that here there's some flexibility going on, some protein dynamics. And actually, when Calmodulin is binding its partner protein, which can be simulated by adding a target peptide, so it's just a small oligopeptide sequence in solution to the protein will close onto the peptide, and so you see the conformational change is dramatic. But actually, you can show in solution that this extended conformation has been crystallized, but you can actually show in solution that it's very flexible, and so it's likely actually that even in the absence of target peptides, Calmodulin in solution will sample conformations that are more compact than this X-ray structure is suggesting. So the idea is to use both pseudocontact shifts and residual dipolar couplings together to investigate the flexibility of this system. Why are they useful in combination? It's written here. Pseudocontact shifts, I forgot to mention that in this system we are able to selectively replace one of the 4 calcium ions with one of these paramagnetic lanthanide ions. So we can have a system where only one of the 4 is paramagnetic. So what happens is that pseudocontact shifts, because they are metal distant, they dependent on the distance between the nucleus and the metal ion, are very small when you go to when the metal is bound to one domain, for example, the N-terminal domain, you get significant shifts for the atoms in that domain, but the others are too far away, and so you get very small pseudocontact shifts. Residual dipolar couplings instead do not depend on the distance, so in principle they should be the same in both domains as magnitudes, let's say, if there were no dynamics. On the other hand, pseudocontact shifts in this domain, which we assume is internally rigid because we are only interested in the inter-domain flexibility, provide an easy way to measure these, to determine these magnetic susceptibility and isotropy. So we combine them by using the pseudocontact shifts in the N-terminal domain to compute the anisotropy, and then we apply that to predict the residual dipolar couplings in the domain that is not harboring the metal ion. Experimentally, what we see, so what we see experimentally, is so if the protein were rigid, as I said, you should get the same distribution of residual dipolar couplings in the two domains. But if it's mobile, because, as I mentioned this morning, residual dipolar couplings are very sensitive to dynamics. So this part is oriented, but the other, the red part, which is harboring metal, is orienting with respect to the field. But this other part is mobile. It's not completely, let's say, the motions are not completely free with respect to the first domain, but there are motions. And so what you see, it's an effective averaging of the residual dipolar couplings. So in the presence of dynamics, you get a significant spread of residual dipolar couplings in the domain where the metal is bound, but they become much smaller in the second domain because they are dynamically averaged. And you see the averaging depends on the metal and so on. I will just try to. So what we do in this maximum occurrence calculations is we produce a number, or we generate a number of different possible conformations, and this is done using one of the programs of the suite that the SACS people developed, where they have this, if you remember from yesterday, they have the program where they have the fixed domains and they compute the linker in between them. You can actually ask the program to randomize these linkers, and so you get a mapping of all the possible distribution or possible relative configurations of the two domains with respect to one another, given the number of amino acids or dummy residues, as Al was calling them yesterday, that connect the two. So if you generate several thousands of these different poses, you can kind of map the configurational space of one domain with respect to the other. And then for each domain we can compute, because we know the for each domain we can compute from these two formulas, because we know the magnetic susceptibility and isotropy. For each of these conformations we can compute the pseudocontact shifts and the residual dipolar couplings. And because we know that the protein is jumping from one conformation to from one configuration to the other, where the domains are moving one with respect to the other, what we produce is ensembles that contain 10 to 50 different configurations, which on average satisfy the distribution of data that we obtain. So in other terms, because we know that the residual dipolar couplings are an average measurement over all the configurations spanned, and that's why actually they get reduced in their dispersion, we can try to reconstitute this average by computing for all possible conformations in a large library of different alternate configurations. We can compute what the data would be if the system were rigidly in that configuration. And then we mix them up, so we construct ensembles of configurations with different weights for each configuration, whereby by computing the weighted average of all this data, we get to satisfy the experimental restraints. So what I would suggest is, since it's time we go for coffee, and then I just have a couple of slides to show how this is implemented, and then I will walk you through the web interface. The basic idea is that we can use a combination of the pseudocontact shifts and these residual dipolar couplings to build ensembles of conformations that, as an average, satisfy the data. And we only do this as an average because if there's dynamics, by NMR we are measuring the average residual dipolar couplings. So if we know, I mean, all the purpose is to investigate the flexibility, so we know that the system is flexible. It doesn't make sense to try and represent it as one conformation, but we just need to do this averaging thing. And this is very well documented in the literature, actually, where people have developed different averaging or ensemble building strategies to do this sort of analysis. But of course, this is special because we're using this paramagnetic stuff that is local specialty of Florence, let's say. So what does this term maximum occurrence mean? What we do is we don't build one single ensemble. This is also another peculiarity of this approach, but we build several tens or even a few hundreds of different ensembles, in which are all centered around one conformation. So basically, at the beginning, we generate a large pool of possible different configurations. As I said, this is done using this the software from the SACS people, where we model the linker. So if you model the linker randomly, you basically end up with a number of different configurations. The structure of the domains is fixed, structure of the linker is changing, and so you get these different shapes. And we generate several thousand of these, typically 10 to 50,000 different configurations, so this pool is really large. Then we select one randomly, and then we start selecting other structures from the large pool, and we keep the weight of this red structure fixed in the averaging, so it's a weighted average. We keep one weight fixed of the initial configuration, and we optimize the weights of all the other configurations in the ensemble, which typically has a fixed size, 40 to 50 structures in the ensemble, and we calculate the target function. So we optimize the weight in a way such that the agreement with the experimental data is maximized, or the residual errors are minimized. Okay, the target function is just the sum of square of errors, as usual. And then we keeping this structure fixed, we try to increase its weight, and we select a different ensemble with this weight fixed but higher than before, and we recalculate the structure, and we keep increasing the weight of this single configuration until it gets larger than a fixed threshold. And again, the fixed threshold, of course, has some degree of being arbitrary, and that's probably the most difficult part of the job. And we do, so this is what happens with one structure around which we will build an ensemble. And as I said, we actually generate several hundreds of these, or let's say a few hundreds of these, 300-400, meaning that we just pick another structure at random and we redo the same trick and again until 300 or 400 structures, and we build all of these curves. So what you will get is that maybe the red structure is, let's say, stable or can be inserted in an ensemble with a weight up to 40% before the target function gets exceedingly high. And maybe there's another structure which just can, let's say, sustain only very small weight, and then it's already gone away, and then there can be some intermediate situation. So what we map basically is that there are configurations like this which have a very low likelihood, and configurations such as the red one which have a much higher likelihood. Of course, this is a heuristic. We are not really providing a systematic search in space. We just try to generate enough conformations and enough ensembles within the domain of configurations that we hope to have mapped the space enough. Okay, this is the target function. And at the end, this is a possible representation that we generate where each structure, sorry, this is the domain where we put the metal, the red ball here, and the other domain can, in the random pool of configurations, can span this entire space, and the second domain is represented by a small Cartesian three-axis centered in the center of mass of the domain. So you see that's the span, that, and this random is just the total. By the way, some of the calculations we started before are finished and some did not start yet. So you see, this is a demonstration of what I was telling you before. Don't expect them to become all available at the same time. Oh, let's see if I can go back to... very happy, but actually I can use my own account now. Wow. Okay, sorry, too many passwords. So the MAXOCC calculations is same interface as before. This interface is actually divided in two steps, because I told you we first generate these 50,000, for example, conformations, which is representing for us the entire configurational space. And then for each of these, we have to pre-compute the pseudocontact shifts and the residual dipolar couplings. So that's the first step, this prepare input thing. Because each individual calculation just takes up 1 or 2 minutes, but since we want to do 50,000 in principle, then it becomes long enough that it's useful to send that to the grid as well. So we have two different different blocks. So first we send this preparation calculation to the grid. We retrieve all the profiles for each of the conformations in the ensemble, and then with that we can run the analysis. That means building up these ensembles. So for this you actually have the data available, but let's just do it. The data are available from that URL, but since we've had problems already, let me just show that for you. I also forgot to mention that for each portal you will find one or more tutorials also on the website. So if you actually go through the support center, Alexander probably mentioned this yesterday, you can go to tutorials, NMR, and then, for example, you can take the MAXOCC tutorial. So, calculations using the web portal, and here you will also find the example data to run the tutorial. They are linked here. So you don't need to download them now. If you don't want to run it in the afternoon, you can just go to the tutorial and there's the link to the data somewhere here, example data. And this is true for all of the portals actually, and some have multiple examples. I think there's also a tutorial, for example, with protein ligand complex with amber. So going back to the calculation here, you need to upload the sequence, so the sequence of the entire protein, and then you have to tell the program how many domains you have in the protein, because it will fix one domain or the other with respect to the other. So it needs to know how many units it has to split the sequence in. So now we are going for 2 domains, and now you have to input the PDB structures of the 2 domains. Right? These are needed because the program will keep these structures fixed, and it actually uses them to compute the pseudocontact shifts and the couplings, and it will move around just the linker region. So by looking at the sequence in the 2 PDB files provided, with respect to the sequence of the entire protein that you gave as the first file, the program will automatically guess which are the residues forming the linker region. So it's by subtraction, okay? You have some residues in the first PDB, some residues in the second. This is the entire sequence. What's missing here is by default the linker. And now you have to, so as I mentioned, now we are just generating these 50,000. No, because it's arbitrary. Actually, in principle, it depends on, because you compute each ensemble in an independent manner. So this is just how much you can raise this weight. So it doesn't really mean that the structure will exist 40% of the time in solution, because it actually depends on how you select, but this is random, the other members of the ensemble. So it's just a relative scale in practice. You know that there are some structures that are just more likely, and some configurations, that are more likely, and some configurations that are less likely. And you have a relative scale, but we cannot say these are neither true probabilities, in the sense that then you integrate over all the space and you get 1, or you cannot compute energetics out of this. So it's just relative terms. It becomes useful. I don't know if I still have the example, I throw it away. If you have free Calmodulin and then you bind the peptide to it, then you see that the space, of course, in principle is the same, because it's just driven by the linker length. But the heat map can change a lot, because now you are restricting the motions. So you get narrower distribution and maybe more centered in a specific part of space.
But since they are relative, it's supposed to possible to get
Yeah, we are not basically computing that. We are just saying that there are states that are more likely and states that are less likely. But as far as if there's 3 states or 4 states, or we cannot even guarantee that if this one is 0.4 and this one is 0.25, maybe in real life this is more populated than the other. That's something that's just not in the method to compute. Of course, you can integrate SACs, because we are talking about things that are determined by the length of the linker. But then if you map these maximum occurrence values, which are the values where this curve crosses your arbitrary target function limit, you see that there are some configurations that have large values and some, so they are hot in this map, and some other configurations are cold, so in principle they are sterically allowed, but their chance of occurring in the solution is very low. What is the unit? They have different shapes. Of course, the shape doesn't change really dramatically because you have 2 domains. You can put them close, you can put them farther away if the linker is more or less extended. Sorry, or they can be in contact like this. So many of these configurations will actually have similar shapes from the SAC's point of view. So the real usefulness is probably to distinguish between, let's say, elongated configurations with respect to compressed. But then if it's closed like this or like that, as Al pointed out yesterday, probably it's not really in the SAC's data. But you can see them. You can see that better from the pseudocontact shifts, for example. So basically, we can put everything together, and that's in the portal. This is telling you why the combining the different data are useful. But I guess now it's really too late to go in that detail. So this is the kind of profile you get for 200 or 300 configurations. So you see that there are configurations that go very high immediately and others that stay flat until you reach relatively high values. But as I was discussing, this is just a relative scale. So you can get different configurations and you can generate these plots easily. That's actually one of the output of the portal, and you map this cloud of possible configurations with the different colors. You can compare to, you can introduce selected configurations. For example, to take free Calmodulin, you map the crystal structures or the extended, so the closed, crystal structures, peptide complexes, or the extended crystal structure. You see they are all cold. So if you take free Calmodulin in solution, it is in neither, or it's not spending the majority of its time in neither of these configurations. So let's have a quick look at the port conformations, for example. The reference domain is the one where the metal was bound to. So in our case, is the N-terminal domain. And then this is actually a parameter. These are all parameters for the for the program that is generating these configurations. So are parameters that determine how large is the space sampled, and typically these are okay. You may want to change this type of native-like or random. It defines what kind of distribution of the linker, of the configurations of the residues in the linker you want to span. So if it's completely random configuration, so basically phi and psi angles of the residues in the linker are randomized completely, or they follow a kind of Ramachandran distribution. This is built in the in the SACS program. And now for the paramagnetics, you can actually you actually have to upload the data. So you upload the residual dipolar couplings and the pseudocontact shifts. These are just text files. And then you have to provide the tensor parameters. I don't go into the format because they are explained on the tutorial, but they are just list of files, okay? The field is relevant to provide the correct magnetic field because you have to keep in mind that these residual dipolar couplings originate from self-orientation, and self-orientation is magnetic field dependent because it depends on the product of this anisotropy times the field. So you have to input the right field and the right temperature. You can also add other paramagnetic effects such as paramagnetic relaxation enhancements. These are, as I mentioned, the paramagnetism increases relaxation rates, and you can actually quantify that. There are specific measurements. These are very popular measurements that you do with spin labels, but you can do essentially the very same measurement from using the paramagnetic effect from metal ions. And then you just give it a name as usual. So basically the important parameters, just to wrap up, is the sequence and the two domain structures. So you have to tell the program which is the structure of the two fixed rigid domains, how many configurations you want to generate. Of course, the larger this number, the longer all the calculations also in the subsequent step, because it will be randomizing from a much larger pool. And then you provide the input data. Let's see if it gets submitted. Okay, here you see actually a single job because this is actually sending so many jobs that we cannot list all of them separately. The list would be too long. So it's a single job which is sending out smaller jobs. So you see like the mother job, whose job is to send other smaller calculations to the grid using in the background. So you only see the mother, you don't see all the small children, which means this thing can actually stay submitted for a very long time because you will see it as S until it has finished sending all of the jobs to the grid. So this thing can stay S for several hours, and that's normal actually, at variance with what we mentioned before, that it should change from S to R in a relatively short time. So after these calculations has finished, and of course I have a pre-computed one, you can actually run the analysis that is building the ensembles. So what is asking you here is to select a project where you stored the results of the input preparation, and then you select this calculation where you have the input data. So it's taking the output that's of the first step that was stored in this project under this name, and now you can start generating the ensembles. So here you basically tell how many structures you want to do the calculation for, and you can actually index them. So if you want to have some specific structure analyzed, such as the X-ray structure, you need to add that to the to this output. So say you put it as the first structure, and then you provide the range. So if you want to calculate 10, you say compute from 1 to 10. So this will take the first 10 structures in the pool of configurations that was generated, and we'll use them to build 10 different ensembles. As I said, the advantage of using an index actually is that if you want a specific structure analyzed, you just add it to this pool of configuration, and then you know that the program is going to pick that. Otherwise, if it's random, you would never be sure what's going on. And then you have to go through a routine, or let's say to iterate this calculation several times, because as you see, there are a number of parameters that can be optimized. Because the protocol for annealing, which is described in the papers, and it's very complicated. It conjugates a number of different steps. So there's a minimization, then by conjugate gradient, then there's some simulated annealing going on, and then it randomizes the ensembles again, and it goes into cycles. So basically get the first random ensemble, and then optimizes the weights, first by gradient minimization, and then by simulated annealing. And then it removes some of the structures from the ensemble, adds new structures, and goes on again, until basically it reaches a condition where the final target function is independent of the ensemble composition. So one of the important parameters to specify, for example, is this one, which is the size of the ensemble. So in this case, you will produce ensembles containing 45 structures. We typically stay between 30 and 50. And then you have these multipliers, because ideally you want the final target function to be in the range that I was showing you on the graphs. Of course, you don't know a priori the range of the target function, because that depends on the also on experimental errors you have. So the idea is that you run this calculation several times, each time, let's say, looking at the first round of results and adjusting these multipliers in order to bring the target function into the range. Is that arbitrary? It's pretty arbitrary, I have to say. Well, if you go into the output files that are produced at the end, you get the residuals from both data sets. So you see if one is having much more violations than the other, then you just reduce its weight, more or less. That's the proportionally. So say you want to get at a target function that's around 0.3 or 0.5, you know that pseudocontact shifts are contributing 0.1, and residual dipolar couplings are contributing, I don't know, 10. You set the multiplier for residual dipolar couplings to 0.0 something, and maybe you can have even 2 for the pseudocontacts.
I'm curious, if I had multiple resonance, can I put them together?
Yeah. You can have multiple. You have to specify. You still upload only one file, but you specify the from which metal you are measuring. In the first step, I had the pseudocontacts and the tensor parameters, but you can actually have in the same file multiple lists of pseudocontacts and multiple tensors, and you just need to tell the program, you know, this is 1, this is 2, this is 3. And at this point, you can actually also add the SACs data. We add them only now because we don't need to pre-calculate them. They are there, and the program that does the calculation of the profile is fast enough that we can just do the comparison on the fly. So there's also the data profile to upload, and you have to instruct the program how many points you have in the profile that are actually to be discarded because they are in these too small, in the region where the angles are too small and the thing is not able to properly calculate the data. You remember that you had to remove some of the initial points. So here you have to tell how many points you discard. And so that's again the same story. This is also a pretty long calculation because, well, now it's only 10 ensembles, so it's not so long actually, but if you are doing 300-400, each is taking up the order of the hour, so again it has to get to be split in several jobs. And it will look pretty much like the same as before, as the others, as the first step. There's only one mother job, and you don't see all the daughter jobs. So just to give you a feeling, then this is the kind of stuff you get out of it. For example, you get a pre-computed plot with all the curves that I was showing you before. And of course, you also get the corresponding numerical data if you want to run. So you get the image, but more importantly, I would say you actually do get the data there. So you can rescale the various contributions as we were discussing just now. And because you see at the beginning, more or less all the structures, when the weight of the conformer is very, very low, you can always find an ensemble that will be happy. So let's say the interesting part of the calculation is when you start increasing the number of the weight of the conformation from which you start building the ensemble. Because if you weight that single conformation very, very low, then you can always build an ensemble that will satisfy the data. The program just needs to pick 50 structures among 50,000, which you can combine and then get the data satisfied. But the trick is when your one configuration is weighted a lot, then the program is not able to do that, and you start having discrimination. But actually, you spend a lot of time computing this because it's always successful. The program spends a lot of time to optimize these ensembles. So actually, after the first round of calculations, when you really know which is the limit value here, which can be 0, but sometimes it's not necessarily 0, because if you have some small systematic error, it will bring your target function to a fixed initial value that's different than 0. You can actually tell the program, look, please don't compute low weight ensembles, and just set the limit that it should go to 5 or 20, whatever. And there was an option in the web portal that allowed you to do that. So you can actually tell the program which limit you should reach on this side, the non-interesting side, and tell the program not to compute that in subsequent rounds, so that you spend less time computing useless ensembles. You get a script to automatically generate this kind of images, and you get a distribution of the different MO values from the program. For example, just to show why it's useful to integrate SACS and NMR data, you see in this example the red distribution is from the small angle X-ray scattering restraints alone, and you see that actually you get fairly large a distribution which centers are relatively large MO values. Because this is a relative scale, you know that if all the values are high, it actually means that these data are not sufficient to discriminate the different configurations. So in this approach, at least, if you only use the SACS data, you are not very effective in discriminating configuration. And as I mentioned, that can make sense because you know that your structures are not going to be dramatically different from one another. If you add the other data, you actually go to much lower values and your distribution becomes more defined. You see a hot region here and a cold region there. And since yesterday we discussed radii of gyration, it's also interesting to have a look at this. The black curve is the distribution of radii of gyration in the randomly generated example, and it's bell-shaped, which is what you expect for a random distribution with this fixed linker. And then the red curve is computed from the ensembles, from the, I think, 300 ensembles where that we generated that equally well fit the data. And you see that you actually get this selection toward either, let's say, an equilibrium between compact structures or actually elongated structures. So we are discriminating against structures which have an intermediate radius of gyration, which is actually in line with what you would expect. The Calmodulin can either have kind of closed structure or tends to be in the extended configuration, and so we can pick that from the data. But if you go look ensemble by ensemble, you actually see very different properties, and that's something that in the literature is not obvious. So it looks like if we are building different ensembles, we are getting the same fit to the data, but the properties of the structures within the ensemble are not always the same. You see this ensemble contains extended structures, but this ensemble actually contains a combination of compact and intermediate structures. So it's true that overall you get a tendency, but the individual ensemble can actually differ a little bit. And this, in our opinion, warrants to the fact, or warrants that one should not look only at one ensemble or a limited number of ensembles, but you actually do need to have a large number of alternative options and then compute averages from those. So it's kind of averaging the averages.